All right, so chromatin. Um, yeah, generic term for any complex of DNA. So we're going to look at euchromatin later and heterochromatin.
Hmm.
Yeah. Okay. Chromosomes are the separate pieces of chromatin that behave as a unit during cell division. Chromatin is roughly 1/3 DNA, 1/3 histone. Do you remember histones?
Yeah, it's supposed to be like complexes of
Think
like
of
things
them
that
as
wrap
like
around DNA.
beads.
Yeah, they're like beads that wrap around DNA and stuff like that.
Yeah. So they're basically made of positively charged proteins.
Yeah.
That's why and DNA is negatively charged, so that's why they bind to each other. And
Yeah.
a group of, I think, eight histones is called a nucleosome.
Okay.
Yeah.
Got it.
Okay. And this is gonna this is how like gene expression works. So okay, DNA interaction with histones and non-histone proteins produces sufficient level of compaction to fit into a cell nucleus. So basically what happens is the DNA wraps around the histones and the DNA condenses. So it goes from like its loose natural form, and as it turns into chromosomes, it condenses.
Got it.
Yeah, and we're gonna like talk about like epigenetic stuff most likely in this chapter, where basically as chromatin gets less condensed, it's open for like DNA polymerase to come in and transcribe it. And as it gets more condensed, that thing gets not expressed. But we can move on to the next slide. So yeah, they're positively charged, highly conserved. Conserved is always going to mean like evolutionarily. Yeah, they bind to and neutralize negatively charged DNA. There are 5 types. Those are the types. These are the core ones. They make up the nucleosome, and that's just the rudimentary or like elementary like DNA packaging unit.
Okay. So what do you mean? What did you say conserved meant before?
Conserved? Oh yeah, it just means evolutionarily, like it's in like every organism basically. So it's been conserved from like the first organisms. Oh, I could see your okay.
Yeah, I'm taking the notes like this.
All right. So okay, so that it's made out of like, well, chromatin's made out of DNA, histones, and then these are the non-histone proteins. So there's they basically they have like functional roles. Like there's there's some non-histone proteins that form the scaffold for the chromosome, like playing a structural role. There's some involved in replication, like DNA polymerases. Some of them work to separate chromosomes, so like kinetochore proteins, remember like the things that attach to like the centromere and get pulled during like anaphase.
Yeah,
And
I
then
got
some
the
and then the largest group are the ones that are active in transcription. So
Okay.
all right, chromosome scaffold. So this is a picture of a chromosome treated with a detergent, which basically breaks it apart. So the scaffold of non-histone proteins is visible in the shape of the 2 sister chromatids. The kinetochore proteins. So they help move chromosomes along the spindle apparatus, so those are like, the red ones there.
Mhm.
Yes. So when stretched out, the DNA in a single cell would be about 6 feet long. The diameter of an average cell nucleus is about 6 micrometers. So, how can something that long fit in something that small? It's compaction, like it gets compacted down.
Okay, got it.
All right. So yes, nucleosome, think of it as like that fundamental unit of the way a chromosome is packaged. And you see at that picture, it looks like beads on a string. Yeah, that's exactly what they wrote too. So yeah, it's formed by the winding of DNA around the histones. And yeah, they resemble beads on a string. Yes, and spacing and structure of nucleosomes affect genetic function. So the string part of the beads on a string are the
Mhm.
exposed DNA. And if it's exposed, it means that it's
Subject to
able
change.
to be transcribed like
Oh,
in
okay.
RNA, and therefore it's expressed, right? And so there are different things, like we'll read about this like probably in the next few slides. Like there's like methylation, which causes it to be more condensed, and then there's like acetylation, which causes it to be less condensed. So yes. Yeah, because like basically, a DNA polymerase can just like attach onto it.
Yeah.
Mhm. All right, so yeah, nucleosome core is an octamer, so 8 total. So I guess 2 of each of these types, and around 160 base pairs of DNA wrapped twice around the core of 8 histones. 40 base pairs of linker DNA attaches the adjacent nucleosome. So that's what we saw in the chain or in the string part of the beads on a string, that previous image. So histone H1, which is not part of like, the nucleosome core, that's that purple thing.
Mhm.
That's the thing that associates with the linker DNA. So like the linker DNA, it wraps around the octamer of those histone proteins, and H1 histone is the one that attaches to the linker part on the outside.
All right.
Okay, X-ray crystallography of a nucleosome. So, that's what they do, to find the structure of DNA. So this is the same kind of technique to look at the nucleosome. So, DNA bends sharply at several places as it wraps around the core histone octamer. The base sequence dictates preferred nucleosome positions along the DNA.
Okay.
The radial loop scaffold model for higher levels of compaction. So chromosomal DNA appears as long loops anchored to a scaffold backbone. The McGraw-Hill like PowerPoints, they always have such bad grammar. I bet they like outsource it or something. Anyway, long, to the scaffold backbone of chromosomes. So that's why you see that you see like a radial pattern, like circular pattern.
Mm-hmm.
Yeah, like it's swirling.
Yeah. Mm-hmm.
All right. Loops of DNA are formed by protein complexes called condensins. So you can imagine what that means. Causes it to condense. So 5 protein subunits constitute condensin. They exist as rings that form around the nucleosome studded DNA, where chromosome passes through the ring twice to form a loop. So that's basically the like red and yellow thing.
Mm-hmm.
Yeah.
Oh, God.
And you see how from left to right, it's like tightening or the loosening like the overall like kind of like noose.
Yeah.
Okay. All right. Super. So this is like a, they're just talking about different like models for packaging. So ultimately it turns into like a supercoil. So I guess the structures underlying that are unknown. One of the possibilities that's supported by electron microscopy is a superhelix of 100 angstroms nucleosomal chromatin compacted into 300 angstrom into a 300 angstrom fiber by supercoiling. So in that picture, you have 100 angstrom fiber, and okay, that's a superhelix, and then you see it as a 300 angstrom fiber, and it gets that way by supercoiling, like it keeps wrapping among itself, like tighter and tighter.
Right.
Yep, this is a karyotype, and anytime you see a karyotype, it has to be metaphase, because that's the only time these chromosomes are lined up like that. So okay, stained with GMSA or whatever, have alternating bands of light and dark staining. Oh yeah, that's the interesting thing. So like you'll see like that kind of separates like euchromatin from heterochromatin, the like banding pattern. So okay, each band contains many DNA loops and ranges from 1 to 10 megabase pairs in length. Banding patterns on each chromosome are highly reproducible.
Got it. I was searching up what GMSA was. It's a nucleic acid staining.
Yeah, you're good at lab.
Huh?
You're good at lab.
Oh no, I just looked it up.
Oh, okay.
No, I won't take credit for that.
All right. So you kind of remember like the whatever centimorgan stuff.
Yeah.
So that was using like basically like math to kind of like figure out where like genes are. But we can also look at banding patterns. So of course, like like the short arm is the P arm, long arm is a Q arm. Yeah, and then light and dark bands are numbered consecutively. So yeah. Oh, and then there's like, I guess, location for genes for color blindness. And you notice it's on the X chromosome, right?
Yeah.
So that's why, I believe it's a sex-linked trait. So mostly men have it.
Yeah.
Yeah, fish. Okay. So that's what they use to characterize genomes. So it depends on hybridization between metaphase chromosomes and a labeled DNA sequence. So chromosomes, they're spread apart on a glass slide and denatured. So, anytime you denature something, you could denature, DNA into, you're basically letting it kind of like break apart into smaller parts.
Mm-hmm.
So like DNA could be denatured into like, single strands. Chromosomes can be denatured into DNA. Like a tertiary structure of a protein could be denatured back into its like secondary and primary structure.
Okay.
Yeah. So they denature it to make them single stranded. And then they label a DNA sequence with a fluorescent tag, like basically, something that when you shoot some light with a certain wavelength at it, it's going to excite it and it's going to shoot it out with a certain color of interest. So yeah, it appears as yellow there. The probe hybridizes to chromosomes at complementary regions.
Got it.
Spectral karyotyping is a variation of fish. So that's just pretty much the same thing, but they're just using a different fluorescent dye for each chromosome to tell them apart.
Yeah.
So that's how they can, when they make the karyotype or whatever, they can tell like, this is chromosome 11, this is chromosome whatever 15 and stuff.
Okay.
Yep. So here we go. Heterochromatin, euchromatin. So heterochromatin is very condensed, which means it's inactive transcriptionally because like a protein needs to attach the DNA to transcribe it into RNA, right? But if it's too condensed, like, the histones or the nucleosomes rather are too close to each other, there's no room for, like RNA polymerase to like get in there and cause it to transcribe. So that's the area of where a gene is not expressed. So they're the darkly stained regions of the chromosomes. Constitutive, which means that it's condensed in all cells. Facultative. Oh no, sorry. There's a difference between constitutive, which means that it's condensed in all cells, and facultative, where it's condensed in only some cells and relaxed in some other cells, aka euchromatin. So, for example, position effect variegation, X chromosome in female mammals. All right. And so euchromatin is when it's loose. And since it's loose, the DNA is exposed, right? And a transcription factor can just, attach onto it and cause transcription of a gene. And those are the lightly stained regions of the chromosomes.
Got it. Okay.
All right. So promoters are hidden when they're wrapped in nucleosomes. So, you have your nucleosome there, and that promoter is wrapped around a nucleosome. So there's no way for, like a polymerase to attach to it and to make it, I think like the real one is underneath, but that one is just like larger for some reason because it's there in the back, but it seems like there's more stuff. Yeah. Okay. Yeah. So, exactly. So basically transcription factors bind to the enhancers, and recruit chromatin remodeling proteins. So that causes basically that part of that of the chromatin to like relax and exposing the gene promoters. That's what they wrote, like removing or repositioning nucleosomes. So that makes it so you see where that promoter is now in for B, it's exposed, and then things can attach to it and turn it into RNA, which means it turns into like its actual gene product. And then for heterochromatin, they're really tightly packed, so we can also say that it's silenced, like the genes there are silenced.
Okay.
Okay, this is an example of position effect variegation in Drosophila. So the white gene, the white gene is normally located in euchromatin, right? The loose type.
Yeah.
And chromosomal inversion can happen. It can result in the white gene being located adjacent to heterochromatin. So the part that isn't expressed. So, we have basically the wild type there, and part of it is euchromatin with that gene, right? And let that W gene, and then RST, whatever that is. And to the right, it's heterochromatin, and then it inverts itself. So when so inversion is when the chromosome inverts itself. So when it inverts itself, you basically have, the white gene, like basically closer to the heterochromatin. So what does that mean? Well, it means that it gets silenced basically. Because
Okay.
up top, it's expressed in all cells, so it gives off a red pigment, but when it gets close to the heterochromatin, it gets silenced. So there's no pigment, but it's not completely silenced because it's not literally inside of the heterochromatin, it's just adjacent to it. So in other cells, it is, expressed.
Got it.
Gene silencing can be caused by spreading of heterochromatin into nearby genes. Spreading can occur over 1,000 kilobases of chromatin. Heterochromatin spreads further in some cells than in others. So, and that says it spreads linearly, so, in a line. So we see like that first rearrangement where it doesn't invade another gene, like the RST, or they're separate or whatever. And then in the white smooth sectors or whatever, that white gene is in the chromatin, right? I mean, the heterochromatin. So it gets silenced, but the RST gene is still active. And then I guess white rough sectors, both of them get, spread to the heterochromatin, so they both get silenced. And then let's see, red rough sectors never observed. So I guess that's just when the RST is still stuck in the heterochromatin, but the white gene is not. And that pattern is never observed though, because it's spreading linearly. So you're never going to basically have like the order is always going to stay the same. So it's never observed. Therefore, that's how they know that it spreads linearly without skipping genes, right? So like, basically, like, I don't know how to put this. It has to basically always be like in order, I suppose. Like, whereas, the white gene is closer to the heterochromatin than the RST gene.
Okay.
So you'll never see, and you can see like, the white gene getting closer and closer, and eventually the RST gene gets closer and closer, but you're never going to see like the white gene not in the heterochromatin anymore and the RST gene still in the heterochromatin. So that last part, even though like that diagram says that it's like that, it's not observed.
Got it.
Mm-hmm.
Okay.
Okay, so this is how they identified the molecules involved in the formation of heterochromatin. So genetic screens in Drosophila were used to identify genes involved in chromatin modification. So they looked for changes in the amount of the position effect variegation. They found so mutations that enhanced the formation of chromatin made eyes more white. Mutations that suppressed heterochromatin made the eyes less white. So even though it's called a white gene, if it's expressed, it appears red.
Okay.
Right? So, and if it's not expressed, it appears white.
Got it. Okay.
Yeah, it was like in that picture from like, a couple of slides before. So they just basically, so this is how they found it out. So they basically just found mutations that made it more heterochromatin, resulted in eyes that are more white. And and less heterochromatin means that I'm sorry. If there's more heterochromatin, yeah, the eyes are going to be more white. And if there's less heterochromatin, they're going to be less white. And by the way, what they mean by variegation, if you can go back to that slide, I think it's 2 up. So what they mean by variegated, like that word itself means it's kind of like remember like incomplete dominance. So like complete dominance is like, let's say for a flower, either red or white. And then codominance is when they're a mix of the two, like being like pink.
Yeah.
But codominance. What was I gonna say? The other one? Damn. I just said it. Incomplete dominance, I think.
Yeah, like hybrid vigor is.
Yeah, so that's where it's not going to be red, it's not going to be white, and it's not going to be a mix like codominant. You'll have some areas that are going to be red, some areas that's going to be white.
Yeah.
So variegation means like the same thing. It just like if you go one slide lower, like to the next one, you see that in the eyes. So it's never just like all red or all white. It's like, it's variegated. Okay.
Got it.
Mm-hmm. Okay, so yeah, then the last part of the barrier elements are DNA segments that block the spread of heterochromatin.
Okay.
All right. The histone tails can be modified with chemical groups. So this is where we're gonna talk about methylation and stuff like that. Ubiquitation usually means that it's going to be sent to be killed or destroyed. So we can use that as platforms for modification, like the tails of the histone. So keep in mind, these aren't parts of the DNA, they're parts of the protein.
Okay.
Yeah. So they're basically the tails of the histone.
Okay.
And yeah, they're going to play a role in epigenetics, which we'll see. All right, so here's acetylation. So okay. So something called a histone acetyltransferase, whatever, it's an enzyme. It adds acetyl groups to histone tails. So an acetyl group is basically like a carbonyl group with let's see if I can find a picture. Whoops. Down. Like, I wonder if I could send pictures through Zoom. I'm going to try. Oh. All right. All right. I can't, but basically just it's carbon with a double bond to an oxygen, like a carbonyl, and then another carbon, like a methyl group.
Okay.
Mm-hmm. So, okay, that's an acetyl group. So it adds that group to the tails of the histones. And what it does is it prevents the close packing of nucleosomes. So it makes it, more relaxed and makes it more, closer to euchromatin. And that always basically makes whatever the genes more expressed. And you could reverse it with, just by deacetylating it.
Got it.
So I guess like in that diagram, you see heterochromatin, and then it gets acetylated, like after that barrier element or whatever, the AC stuff, those are the acetyl groups, and it makes it euchromatin. It's more spread out. Make sense?
Yeah.
Okay. We're going to see methylation next, I bet. Yep. So histone methyltransferases, they add methyl groups, and that's just a CH3. The effect depends on the specific amino acid modified. So adding it to the lysine, the 9th lysine of the H3 histone makes it more like heterochromatin. So basically makes it more condensed. So basically going from the right in that diagram to the left. So that's why on the left you see the M's.
Yeah.
Those are the methyl groups.
Okay.
Yeah, and then the HMT, that's the histone methyl transferase, and it just basically takes a methyl and it attaches it to the histone. The HAT, that's the histone acetyl transferase, and it attaches the acetyl group.
Got it. Got it.
Mm-hmm. Okay, so bar bodies, like, you know, that's like, you remember, like since like men and women are supposed to have equivalent genetic material, so some X chromosomes would like deactivate, or, you know, for females, and that occurs through the formation of heterochromatin. So these bar bodies, they're examples of facultative heterochromatin. So yeah, it was called dosage compensation. So like, you know, like an X-linked gene in a female with an XX chromosome and a male with an XY chromosome, like there should be double the genetic data for the XX chromosome, right? But in order for it to be equal, one of those X chromosomes gets deactivated.
Got it.
And a deactivated X chromosome is called a bar body. And like this happens, like, you know, in like individual cells. Random inactivation for all except one X chromosome in each cell. The inactive X chromosome, called a bar body, appears as darkly stained heterochromatin masses when you observe somatic cells in interphase. So yeah, this only happens with somatic cells, not with sex cells, because of course with a sex cell, like an XX chromosome just means that you're like female, so you can't have it like inactivated, but just in regular cells. Yeah, that's what, you know, what somatic cell, it says that. Yeah, but yeah.
Yeah.
Mm-hmm. Yep. Okay. In a very early human embryo, both X chromosomes are active. In humans, random X inactivation occurs around 2 weeks after fertilization. Some cells have maternal X chromosomes inactivated. Others have paternal X chromosomes inactivated. All cell descendants will therefore have the same inactive like X chromosome.
Okay.
X inactivation is initiated by the expression of the exist gene, or exist gene, however you pronounce it. So, you know, exist or whatever, you know, stands for whatever X inactivation specific transcript. It's expressed on the inactive X, but not the active one. The RNA for it, though, is large, non-coding, and cis-acting regulatory RNA. So it's a type of non-coding RNA that works to bind to the X chromosome that it was expressed from, and it causes histone modifications like methylation and deacetylation that causes it to condense. And if it condenses, it means that it doesn't get expressed.
Got it.
Okay. Rate of DNA synthesis in human cells is about 50 nucleotides per second. And so it would take 800 hours to replicate the human genome if there was only one origin of replication, also known as ORI. But we have a ton of origins of replication. So most mammalian cells have around 10,000 origins. Many origins are active at the same time. Accessible regions of DNA that are devoid of nucleosomes. So AKA, so like, origin of replication, right? It needs complete access, like the DNA polymerase needs complete access to it, right? For replication. So these are just regions, that won't have the nucleosomes because that blocks it. And a replication unit, which they call a replicon, is basically DNA that's being replicated in both directions from one origin, just, bidirectional.
Got it.
All right, nucleosomes are disassembled and reformed during DNA replication. So basically, like, in whatever interphase, like, when in the S phase or whatever, when they, DNA synthesis occurs, they're packaged as nucleosomes. So it goes from not being packaged into nucleosomes to when synthesis starts, within minutes of it, it starts to get packaged into nucleosomes. New nucleosomes are composed of recycled and new histones, and chromatin is open to histone modification after just after replication. So in that diagram, you have the old parental histones, like doesn't really mean it comes from parents, and in that replication fork, replication starting, and we have new H3 and H4, histones added to the mix, and new H2A and H2B histones up there. So basically, you see how it's like the old parental histones are disassembled, and then the new ones are like reformed.
Okay.
Mm-hmm. All right, telomeres, they're the caps that protect the ends of eukaryotic chromosomes. So they basically consist of specific repetitive sequences, not genes. They're species specific, so it depends on the species. So for humans, you'll see the TTAGGG repeat. So like our telomeres will just have that like repeated over and over again. And then it's that for tetrahymena, whatever that is. And yeah, there are 250 to 1500 repeats within with a variable number between different cell types. So it depends on like, what cell, like where it comes from in your body. And what they do basically is they prevent the chromosomes from fusing to one another. Because if you think about it, like each chromosomes, the ends are going to have the same repeats. So they're not going to attach to each other, right?
Yeah.
And also to maintain the integrity of chromosomal ends, because each time your DNA gets replicated, the telomeres get a tiny bit shorter.
Got it.
All right, replication at the ends of the chromosomes. RNA primers used during DNA replication are removed, leaving RNA primers length of DNA missing at the 5 ends. So it's okay. The RNA primers, they're gone. So when this happens, right, if there wasn't a special mechanism, the DNA would be lost each time, it replicates itself, right?
Yeah.
Yeah. So then it's going to like explain why. So telomerase is basically the enzyme that works to extend the length of the telomeres.
Okay.
So kind of like, if you can like define aging as, because you could define aging that way, because, each time your cells replicate to make more cells, the telomeres get shorter. So as you age, your telomeres are, going to get shorter and shorter and shorter. But telomerase, that enzyme, like adds to it and makes it longer. It's actually like implicated in like a lot of cancers.
Yeah.
Having like over overexpressive telomerase that just keeps it going. That's why like you have like, like the HeLa cell, like the immortal cell stem cell lines.
Ah, okay.
Yeah. HeLa cells, like, they're named after like Henrietta something, like this woman with cancer, maybe in the 40s or something. And we
Oh,
still
Henrietta
have,
Lacks.
what's that?
Yeah. Henrietta Lacks.
Yeah. And we still have, her cells. They're immortal.
Yeah.
Yeah. Telomerase activity and cell proliferation. So yeah, basically the levels of telomerase and cellular lifespan varies between different types of cells. So most somatic cells have low expression of telomerase. So and telomeres shorten slightly at each cell division. After so senescence, that's aging essentially. So senescence basically occurs after less than 50 generations in culture. Now germ cells, sex cells, stem cells, and tumor cells have high expression of telomerase. So each time it divides, instead of getting shorter, its length is maintained. So it doesn't age. And you could even call that negligible senescence. Kind of like, how like, those animals, can live like forever, practically, lobsters and stuff.
Yeah.
Greenland shark, turtles.
Yeah. Okay.
Yeah, telomeres maintain chromosomal integrity. There's a proteins called shelterin, and they basically bind to the telomeres and they fold the DNA. So what that does is, so there's always like these nucleases, like just in the cell. There's also ribonucleases, which, so like a nuclease is just going to break a nucleic acid down. Ribonuclease is going to break down, RNA then. So yeah, so these proteins, they, fold the DNA of the telomeres, and yeah, it protects it against degradation by those enzymes. And it also protects it against, so this is like, with chromosomes not having their ends joined, because they loop around each other.
Oh, okay.
Yeah. Artificial chromosomes, constructing yeast artificial chromosomes. They require 3 key elements: centromeres, telomeres, and origins of replication. So you can see there, telomeres obviously at the ends. Centromere, generally it's going to be in the middle, but of course it depends. There's like, those like, forgot, like p-centric or whatever, mid-centric. You remember what I'm talking about?
Yeah.
Where it's like sometimes closer to the top, sometimes closer to the bottom. Metacentric, I think they're called. Metacentric, and then acrocentric. Yeah, remember those?
Yeah, yeah.
Okay, so so the centromere, it's not always going to be dead center, but it is going to separate like the p arm from the q arm and stuff. So okay, that's a centromere, and then we have a gene TRP1. I guess some foreign DNA that you include into that when you make the artificial chromosome, and whatever, like an origin of replication. So let's see. Consequences of deleting key elements. So if you get rid of the centromere, and if you get rid of the telomere, you're going to have errors in replication and segregation. If you don't have the telomere but have the centromere, you could still wait, what? Okay, sorry. If you don't have the centromere nor the telomere, it will, when it replicates, there'll be errors in segregation, like when the chromosomes separate. Now, if you have the centromere but no telomere, when it replicates, it's going to degrade. If the if it's still linear as in like like generally speaking linear, but like unless it's whatever, a plasmid or something. But yeah.
Got it.
Because yeah, if it was a circle shape, then there isn't any telomeres, right? Because there's no ends.
Yeah.
Okay. So all right, yeast fused chromosome consists of a giant 11.8 megabase fusion of all 16 normal yeast chromosomes. So they took all these 16 normal yeast chromosomes, they fused it together into one. Synthetic chromosomes differ from artificial chromosomes. Synthetic chromosomes are entirely man-made, whereas artificial, you're piecing together actual real like chromosomes from yeast in this case. So there's a difference there between artificial and synthetic. Synthetic chromosomes include all genes present on corresponding, this grammar is so bad, on the corresponding yeast chromosome, versus YAC, the yeast artificial chromosome vectors. They usually only have a single protein coding yeast gene. And fusion synthetic chromosomes help scientists study minimal genome components that support life.
Okay. Oh, that's it.
All right. All right, I guess we're going on a macro scale now, because we're talking about, population stuff. So yeah, beyond the individual gene and genome.
Okay. Oh, this test isn't on Blackboard. It's going to be on Connect.
Okay.
Like the thing that we were doing worksheets on and stuff.
Okay.
Yeah, that's like where it's going to be on.
All right.
We haven't gotten a new worksheet in a while. I've been kind of hoping for one, but we didn't.
Oh, okay.
We were supposed to be getting them weekly, but they just stopped.
You can like email your professor and be like, yeah.
Oh, people already did. They were just like, oh,
so I have good practice for the test.
They don't. We've already asked.
Okay.
They're like, oh, we're just giving a break. I'm like, okay. It's like,
all right. Yeah.
Yeah.
Okay. All right. It's gonna be this stuff. This stuff. Harvey Weinstein law. It's gonna be like the PQ, like equation.
I love how these people are like falling that it's not open book. It's obviously open book.
I mean, they don't give you time for it to be open book, remember?
Yeah.
Yeah. So okay. But like this stuff, unlike that last stuff with tetrads and stuff, I've like learned before, so we should be good.
Yeah.
But anyway, population, right? It's a group of
Mhm.
interbreeding individuals of a single species living at the same time and place. Gene pool, total of all alleles carried in all members of a population. A sample, number of individuals, a number of individuals used to make inferences about the entire population. And then there's monomorphic and polymorphic. So one allele or a variant of that allele, or more than one allele or a variant of that allele. All right. Phenotype frequency are the ones, proportion of individuals in population that have that particular phenotype. Genotype frequency, same thing, but with genotype. So here's all right. So gene with 2 alleles, capital A, little a, a population of 20 individuals. Now they're saying that 12 are homozygous for like capital A, and 4 are heterozygous, right? Capital A, little a, and then 4 are homozygous, little a. So the frequency, you just straight up take, you just do like, like out of the total, right?
Yeah.
Yeah.
Got it.
Now, the allele frequency. Now, so in 20 people, let's say that there are a total of 40 alleles, right? That's usually if the case, if there's whatever, 2 letters or whatever. So yeah, the 12 like homozygous capital A individuals, since they like, whatever. Before we're talking about the gene, but this is the allele, so you're basically just doubling it. So that means that there are 24 capital A alleles for that other the heterozygous group or whatever. There's 4 capital A alleles and 4 little a alleles, and then it goes like that. And so then you can do it like then you can figure out the frequency that way. So this is actually nice because they're kind of like, without just telling you like p plus q is equal to 1, and then p squared plus 2pq plus q squared is equal to 1
Yeah,
also.
it's actually putting it with numbers.
Yeah, it kind of explains like like I guess when you would use that and when you'd use the other thing, but I think it'll like come to that like specifically, but yeah. From genotype frequencies to allele frequencies. Okay, so yeah, here we go with the one thing. So like the allele frequency, that's why you'll see like the p plus q is equal to 1, right? The 0.7 plus 0.3.
Yeah.
And then for the genotypes, you'll have, yeah.
See if this works.
All right, I'll try it. Thank you.
You can use this bathroom only for people.
I know. Okay. I'm tutoring. Close the door.
Okay.
Thank you.
Is it working? Let me know.
All right, okay. Thank you. Okay, so yeah, so for the other one, but Daddy, I'll check that later, okay? Thank you. All right. So anyway, whatever. Next slide. Okay, yeah. Hardy-Weinberg equilibrium. It correlates the two. And there are certain assumptions. These are the assumptions for it that must be true in order for you to be able to use the Hardy-Weinberg equilibrium. Like there has to be an infinite number of individuals in the population. The individuals must mate at random. There can't be any new mutations. There can't be any new migration into or out of the population. and the genotypes can't have any effect on fitness. So they can't, cause someone to die off or something. So basically
Yeah.
you have this perfect, ideal environment, and it's just to see how the allele and genotype frequencies move around in that population.
Got it.
All right. See, this is all right, this is,
Just got a new update with the exam.
Yeah.
50 multiple choice questions. Nothing has to be explained.
okay.
So, it's from 5 to 6:15.
Okay. So I'll show up at
So,
4.
yeah, come to my house at like 4. We'll just make game plan or whatever and open up anything.
All right. Sounds
And,
good.
yeah.
So, okay.
Yeah,
This is a really good picture because Punnett squares don't take into consideration Punnett squares, they're all equal sized, right?
Yeah.
But this is more like a realistic example of it, where the sizes correlate to the percentages.
Yeah.
So anyway, whatever, for whatever population that satisfies all the assumptions for the Hardy-Weinberg equilibrium.
Yeah.
This is the equation for it, basically. So basically, so P represents the frequency of A, the allele for A. Q represents the frequency for the little a. And that's, the formula. And I can write down, because you're also going to see this. So I'll write down in the thing. So P plus Q, plus Q is equal to 1. And so how do you get from there to the thing that they gave you? It's a binomial expansion. So basically P plus Q squared, right? Which is P plus Q in parentheses times P plus Q in parentheses. So you could do FOIL, right, to get P squared plus 2QP. I guess I write 2PQ plus Q squared. Whoops. And it's still going to equal 1. So that's binomial expansion. All right. Kind of makes sense?
Yeah.
All right. Yeah, this is also a great way to look at it. So you see how it's kind of exactly like the SIR model for epidemiology for epidemic,
Oh yeah, the SIR model. That's right. Yeah.
It's the same kind of shape.
The survive infected, and then what's the other one?
So okay, if this was that, the infected is the blue one because it starts at 0, and then it would reach a peak or whatever. And then the people who are susceptible, that's the S part, is the red one. So people who are susceptible.
Okay.
And then the dead or recovered would be the green one. Here, let me SIR, give me logical model. It looks slightly different, but basically it would basically top off, because the people who are susceptible, right? So okay, S susceptible, I infected, R recovered or dead, right? So the people who are susceptible, right?
Start off at recovered. There we go. That's
right.
Oh, sorry. Dead, yeah.
So people who are susceptible start off at 100%, like the red, and it goes down because they get infected. And then the infected would be basically the green. Like, you know, as the people who are susceptible get infected, the green are the people who get infected, and that goes up, right?
Mm-hmm.
And as that happens, the people who are whatever, recovered or dead go from like
Yeah.
zero, that would be the blue one. And so as you know, they recover, that's why the infected would go down.
I see.
So
Okay.
yeah, so that's just, you know, being able to like look at like the graph and being able to see like patterns like that. So it's basically the same thing for, you know, Hardy-Weinberg equilibrium. Like basically, you know, dominant gene, right?
Yeah.
You know, over time, like people who will be homozygous for it will increase. People who are homozygous for the recessive gene will decrease, and people who are heterozygous will increase until, you know, the two cross, and then it'll decrease.
Got it.
Yeah.
Okay. Oh, that's it.
Right.
I guess it was a review.
Mm-hmm.
Okay. So cool.
And then you said you got like a new worksheet or whatever, or an email or whatever.
No, I just had new information on the test. That's all it
Just the
was.
50 question thing.
Yeah.
Okay.
Yeah. Today's session was going to be shorter anyway. Let's see. Since that we're not going to get chapter 24 until Wednesday, probably. We've gone through those, so. Okay.
Why would they send it on Wednesday if your test is on Wednesday?
Huh?
Yeah.
Or maybe that they won't even send it for chapter 24. That's probably the case.
Oh, it's not on the test, right? You're saying?
No, because chapter 24 is on the test, but being
Okay.
that it's like literally vocabulary and stuff, it's not even much else. And we've done Hardy-Weinberg in the past.
Oh, would it be
So-
more about Heidi-Weinberg then?
I guess.
So
And I
like
think it was there for more review. If
they
anything.
probably ask questions because we haven't had any, you know, Hardy-Weinberg questions.
Yeah.
Because
They figure it's 50 questions, so they're gonna ask 10 on 9, 10 on 10, 10 on 11, 10 on 12, and 10 on 13.
mhm.
So
So
and they
yeah.
said second half of lecture 9.
Mm-hmm.
So if it's 74 power slides on lecture 9, that means that like
Mm-hmm.
it would have been from like wherever the first break is here. Part 2, yeah. Once part 2 starts is where
Mhm.
they continue.
So sometimes the Hardy-Weinberg questions are kind of hard, like tricky. Wait, is this
Yeah.
something that we haven't looked at? No, we
No,
did.
we've looked at this. We've done this.
Spisosome right there.
Yeah.
Okay, so why don't you do a Google image search, Hardy-Weinberg questions?
Okay.
Like I sent you one in the chat, like a link to one. Oh, that's annoying. I just meant to send the picture, which I guess you can see if you whatever, just Google image search Hardy-Weinberg questions.
There's a Hardy-Weinberg calculator. What?
Yeah. Yeah. But we have to know, like, you know, oh yeah, so like do like image or whatever. So let's see. Okay. Can you like try to, okay. What is it? All right, what's the
There you go.
frequency? Okay. All right, the recessive allele B occurs with frequency
Occurs with a
0.8.
frequency of, yeah, in a population of crabs that is in Hardy-Weinberg equilibrium. What is the frequency of the homozygous dominant individuals?
Okay. So how do you think you'll do this?
Well, we have a frequency of 0.8, right? So when we want to like create a Punnett square,
No. So you would have to use the P
or we're just
equation.
using P plus
Mhm.
Q.
So that's how you start, right? So that means that P is equal to
Okay.
point. Oh wait, recessive. Okay, so Q is equal to 0.8, right?
Uh-huh.
So P must be
0.2.
yeah. So now they're asking for homozygous dominant. So that's what in the equation?
The 0.2.
Uh, well, well, the so okay, P, but if it's homozygous, it'll be P squared.
Okay. Okay.
So what was P? 0.2, right?
Yeah, P would be 0.2.
So then P squared.
It's 0.4. So A.
0.04. Yeah.
Yeah.
Since it's a fraction, it gets smaller.
Yes, that's right.
All right.
Okay.
Let's look at like another one, because the reason I'm saying that it can get tricky is because sometimes you may not know if to use P plus Q is equal to 1 or the, P squared plus, All right. Frequency of vestigial wings in a population of Drosophila of 50, whatever, is 4%. How many flies would be heterozygous with regard to wing type? Assume, all right, so okay. So all right, so basically heterozygous doesn't really tell us if it's dominant or not. If it's vestigial wings, how many would be heterozygous with regard to wing type? I mean, like, okay, so I'm not really sure if it's dominant or not. So I don't know which one it's going to be, P or Q. Wait, you know what? Wait, I guess it doesn't matter because heterozygous would be the 2PQ, right?
Yeah, that's what I was thinking.
Yep. So okay. So 4%, right? Would be what as a decimal?
0.04.
Good. So let's say that's whatever, P, right? Then Q would have to be like what?
96%.
Yeah, 0.96, right?
Yeah.
And then we just multiply those and then multiply by 2 for the 2PQ part.
Multiply those, then multiply it by 2. Okay.
What do you got?
0.0768.
Yeah. So now you just multiply it by the total number of people in the population because it's a percentage, right? So multiply it by 1,500.
115.2.
All right, wait. We did something wrong. So okay. You did 0.04 times 0.96 times 2, right? And you got,
Well, I got the answer for that, and then I times the answer by 2.
okay.
Right. So hold on. Let me see if I times it by 2. I guess something else. I still got the same answer.
Let's see. 115 or 112.5, you said?
115.2.
115.2.
If you times it by 2, it's 2.30. It's 230. But assume random mating. but no migration, mutation, random birth, or natural selection.
Oh, wait. Okay. Wait. Damn it. Then that means we have to do the oh, you know what? We could still do it. Okay. So because we have to set so okay, do this. I'll write it down.
Got one.
All right. That means because it's supposed to equal 1. What the H then? Well, it's on Reddit. They'll tell you the answer. Let's see what they say. Oh, it's an MCAT question. Yeah, these types of things come up on the MCAT. What do these losers say? Yeah, he's saying he's getting around 110. And he apparently the answer is C. So let's see. Which is doubling of the 240. So it's quadrupling what we got. Let's see. I recall this question. Wait, what? Free and not oh. Used for the expression. Okay. There's a frequency of homozygous. Okay. Well, okay, duh. 4% is describing the homozygous recessive condition. Oh, okay. See, it didn't freaking tell us. Yeah. So okay, that F that question. Because it didn't tell us that it was that the 4% like is the homozygous recessive, aka Q squared is equal to 0.04. You have 100 pet llamas. What is that? 74 of them have curly hair. What is the allele frequency in the population of the 3 genotypes? Okay, so what are you looking at?
Oh no, I was just going on to this thing really quick.
Okay.
Some reason. Saw something.
How's chem?
Okay. I haven't really done anything for it. I've just been leaving it for the summer at this point. Because they gave us the whole summer to do it, so there's gonna be section quizzes and stuff like that. Due May 23rd, that's a lie. That's only temporary. Okay, sorry. Thought I saw someone say that there was quizzes on there, but there isn't. This is the question we were up to, right? Okay, so 74 of them have curly hair.
So
Crossed out
42
6.
homozygous dominant, 32 heterozygous. So tell me what these things are in the PQ stuff.
So on the PQ stuff, we have 100 plant llamas, so that's like out of all of it. So that's gonna be our P. I mean, that's gonna
Uh,
be our Q.
So P is the, you know, the dominant like allele,
Okay.
and Q is the recessive one.
Okay. I got it.
And
All right, so
once they talk,
42 is our
was it?
42 is our P.
So okay, now when they talk about homozygous dominant, then since it's two of those alleles, right, of the dominant ones, it's P squared.
Yeah.
And if it was homozygous recessive, it would be what?
Wait, what?
So like
26, right?
um
Because we have 42 homozygous dominant, 32 heterozygous. Which
oh, I
makes
just
up the
mean like
74.
in terms of like the like from the PQ stuff.
Mm-hmm. Okay, well, dominant is P, so 42.
And that's homozygous dominant, right?
Yes.
So that's P squared.
Oh, okay, yeah, that's right. Homozygous is always gonna be squared. Sorry.
Mhm.
All right, I got it.
Yeah. And then it says 32 heterozygous, right?
Yeah.
That would be which one?
Um,
Like in the equation.
Q? No, Q is recessive. So P squared plus
There's
just
like
P?
that thing in the middle, remember?
Two PQ.
Mm-hmm.
Okay.
So like let me explain this better. So because there's P plus Q is equal to 1, right? And then there's
Yeah.
the one that they give us. So P itself is just the frequency of the dominant like allele, right?
Yeah.
The total amount of capital A, right?
Yeah.
Now, total amount of homozygous capital A, that's going to be P squared.
Okay.
Does that make sense?
Yeah.
Because, yeah, okay. Then, total amount of little a is Q. But if it's homozygous, little a, it'll be Q squared, right?
Yeah.
And then heterozygous will be 2PQ.
Got it. Okay.
Yeah. And then that diagram kind of like indicates that to you. Because if you think about it, right? P like is going to be greater than Q, right?
Yeah.
Like P is 0.7, Q is 0.3, right? So
Yeah.
P squared, right, is going to give you a larger number or a larger area than the box that's like Q squared, right?
Mm-hmm.
So okay, P squared is which box?
P squared.
Mm-hmm.
Sorry. Here I am.
Yeah,
P.
that one. And Q squared? And then PQ? Yeah, so see how there's two of them?
Yeah.
All right. Yeah. Good. So that's like the hard part about Hardy-Weinberg. That's why, they still ask it on the MCAT, because
Yeah.
people have a hard time knowing, like, do I use P plus Q or P squared plus 2PQ plus Q squared? So, okay. So they're telling you that 42 homozygous is dominant. So that'll be the P squared. 32 heterozygous would be the 2PQ, right?
Yeah.
And now they're asking, and by the way, since they're only giving us numbers, we have to use percentages. Like, we're gonna have to do like some math for that, but this is just for like the concept. Then it says, what's the allele frequency? What is that? Allele frequency of curly, capital R, and little r straight in the population. So that's where you use the P plus Q thing.
Got it.
And then for the frequencies of 3 genotypes, right? So like, yeah, the genotype thing is when you use the P squared stuff, but just for allele frequencies, you just use the P and the Q.
Got it.
Mm-hmm.
Okay.
Yeah, let's try like a few more. I want to make sure you can do it. Yeah, maybe like one of these worksheets.
Yeah. All right, here we go.
Oh, nice. All right, this is good. So, okay. What do you think about that first one?
A randomly mating population has an established frequency of 0.36 for organisms of homozygous recessive for a given trait.
So what would
is
that
B
be?
frequency? That's P squared.
Close?
Q squared. Q
Good.
squared. Sorry. What is the frequency of the recessive allele in the gene pool?
What would that be?
So, that would be P, P
Well,
squared?
recessive?
What?
So it's recessive though?
Yeah. Oh, so Q squared.
So, well, Q
Or just
squared
Q.
is homozygous recessive, right?
Mm-hmm.
But the frequency of the allele itself, because that's the genotype, but the allele itself.
Yeah, it's Q.
Good. So, all right. So how would we find this out? Figure this out.
So the equation we can use is,
Well, think about it. If 0.36 is Q squared and we need to find Q, we could just, what can we do?
Q squared square root it?
Yep.
Okay.
Yep. And that's all you do.
So 0.36 square root of 0.36 is 0.6.
Yep.
So the frequency is that.
Exactly. Oh,
Okay.
and then you can, so then what's the frequency of the dominant allele?
0.30.
Mm,
Oh, no. What do I do from then to find the dominant then?
so you found Q, right?
Oh,
Mhm.
okay. So P is just going to be whatever is left after 36, right?
Ooh,
So 64?
whatever's left after Q.
Like it's 64, right? Yeah.
Nope.
Not 0.64?
No, because that's Q squared, the 0.36.
Oh,
And the
okay.
Q was 0.6, right?
Yeah.
And then
Oh, so then 0.4 is the frequency of the dominant?
exactly.
Yeah,
Of the
because it's out of
dominant
100. I got
allele.
it, because it's out of 1.
Yeah.
Uh-huh. Got it.
All right.
Understood.
So yeah, now you see the difference between the genotype, where they say homozygous, but the frequency of the allele itself is the smaller version of that. Okay, so larger version. Even though you're squaring it, this is a funny thing. Even though you're squaring it, it's smaller because it's a fraction.
Okay.
Did you notice that? Like square root of, or like 0.6, like square root of 0.36 is larger.
Uh-huh.
Okay. Next one, they found
Okay,
gene A in 2 allelic forms like big A and little a. And they're saying if the frequency of the dominant allele in the population is 0.7. So, okay, from there, what is that going to be in terms of like the PQ and stuff?
Mhm. All right. Can this be the last question we do?
Yeah, sure.
Yeah. In a large population of organisms that is in the hierarchy. Okay, gene A is found in two allelic forms, A and little a. If the frequency of the dominant allele in the population is 0.7, then approximately how many individuals out of the 100 would expect to be homozygous recessive?
Yeah, so don't worry about that yet. First, tell me what that 0.7 is. Is it Q? Is it P? Is it P squared?
Oh no, it's P. It's P.
Perfect. So
It's just gonna be P. It's not squared
good. And then Q
out of
is
anything. Q is gonna be 0.3.
perfect. So okay, now they're asking homozygous recessive, right?
Yeah.
So that would be what?
Square root of 0.3, right?
Tell me in terms of like PQ and stuff like that.
Oh, okay. Q squared.
Q squared, right? So Q
Yeah.
was 0.3.
Yeah.
So Q squared would just be 0.09, right?
Yeah.
Yep. And then they're just saying how many out of
How many
100?
individuals?
And
It would be
0.09
nine.
is 9%, so 9, right?
Yeah, nine individuals.
Yeah, good. So
Okay.
yeah, the main lesson here is to know, like when you read it, to know if it's P. Like it's easy to know if it's P or Q, but to know if it's P or P squared or Q and
Got
Q
it.
squared. Okay,
Okay.
is that it?
Yeah, that's it.
All right.
So on Wednesday, 4 o'clock,
Yes.
and then we'll make a game plan and then take the test.
Yeah, and if there's any, updates, yeah,
And
let me
anything,
know.
I will let you know.
All
Yeah,
right.
but I've sent you all the other ones that have questions and stuff that I've gone through and then quizzes on. So
That we've gone through together already, or
yeah, and
okay,
I
okay.
can send the rest of the PowerPoints if anything.
All right, okay. Like if you get a new worksheet or whatever, yeah.
Yeah, I'll let you know about that. Yeah.
All right. All right. Take care now.
All right then.
All
Take
right. See
care.
ya.